Right, so I think I'll make a start now, and people can join as I sort of introduce myself and the company. So yeah, for those of you that don't know me, I'm Rachel. I'm the new scientific and technical manager at Vesiculab. So I just joined at the beginning of last month. And before that, I was doing a postdoc on extracellular vesicles, and before that, a PhD on extracellular vesicles. So I'm very much from an EV background, and I've done a lot of nanoflow cytometry experiments before. So today I'm going to be talking about how Vesiculab is helping to advance extracellular vesicle and also nanoparticle research. A lot of our tools are also applicable to other types of nanoparticles, such as lipid nanoparticles and liposomes. And I'm going to be talking through purification through to labeling, analysis and storage, and also preparation of samples for downstream functional assays. And the focus of the webinar is not just going to be on individual products, but on the workflow as a whole. Also, we're going to be recording the session, so you can always watch back or send people that aren't able to make it right now today. So yeah, let me make a start then. So a little bit about us, for those of you that don't know us, we're based in Nottingham in the UK. We're a relatively young company who started in June 2023. And we've now grown to offer a range of reagents, with the main focus being to support and streamline EV workflows, but also other types of nanoscale particles too. So obviously the focus of the talk is going to be mainly EVs. And when we step back and consider the whole EV workflow, there's generally several hurdles that researchers have to overcome throughout the process. EV samples tend to be quite precious, low volume, sometimes highly variable. And so every single step in the workflow matters. And some of the main challenges that researchers face are sample loss during long and laborious purification and concentration steps. Also, when labeling, it's very easy to have excess background noise when labeling with dyes or antibodies. And also instability of EVs during storage can all have a big impact on the quality and the reproducibility of the data. So I'm going to be talking through Vesiculab solutions to these challenges that expand across the entire workflow. And overall, our aim is to reduce the time spent and also reduce material loss, thereby improving the quality of EV samples and data. So starting off sort of chronologically, thinking about EV isolation and purification. So size exclusion chromatography, or SEC, is one of the most commonly used approaches for EV isolation and cleanup. And the principle is relatively simple. So hopefully you can see my laser pointer here. So the column in a SEC column contains a porous resin, and smaller molecules and particles can enter the pores of the beads and so take a longer time to elute through the column, whereas larger particles, such as EVs in this case, are excluded from the pores, and so they travel more directly through the column and elute earlier on. And this leads to separation of extracellular vesicles, in this example, from other smaller contaminants like soluble protein or perhaps free dye or unbound antibody. And the main advantages to this are that it's a gentle isolation method compared to, for example, ultracentrifugation. It's also compatible with many different sample types, and it can give a reasonably good yield overall. But the cons are it's quite slow, and it's also dilutive. And so you have to, in gravity-fed systems, you have to collect multiple fractions, and you thereby end up diluting a sample, typically 3 times, but it depends on the setup. So there's always a bit of a compromise between yield and purity, particularly where the smallest EVs overlap with the larger contaminants, such as protein aggregates or lipoproteins. So SEC is a strong method, but the practical workflow can still be improved in certain ways. And this is where the Vesi-SEC workflow comes in. So it's the same principle as size exclusion chromatography, but it's formatted as a spin column. instead of gravity flow and fraction collection, the sample is simply loaded onto the spin column, centrifuged for 1 minute, and then you elute your purified sample. And again, I've exampled EVs here, but this is very much compatible with other types of nanoparticles as well. So this means that you keep the benefits of conventional SEC, but instead it's much faster. There's no fraction collection and it's non-dilutive. So in terms of particle recovery, we get about more than 90% particle recovery. And there's also 2 formats in terms of the sample volumes that you're working with. So the micro model, the Vesi-SEC micro, is suitable for 50 to 100 microliter sample volumes, whereas the MIDI model is a little bit larger scale, so 0.5 mil to 1 mil load volume. So yeah, the main message here is that Vesi-SEC is trying to make SEC more practical for routine EV and nanoparticle workflows, especially where users want rapid cleanup with minimal sample loss. As well as EV isolation, another particularly useful application of Vesi-SEC is the removal of unbound dye or antibody after labelling. When we're labelling particles, we're often adding dye or antibody in excess to make sure that the labelling reaction is efficient and that any epitopes are being saturated. But then that can become a problem because we then have potentially excess reagent in our samples. And then this free dye and antibody can contribute to background noise, particularly when we're thinking about instruments that are detecting nanoparticles. We're often pushing the limits of detection of those instruments. And it can also make EV fluorescence look brighter than it actually is, or it can mask weak but genuine EV signals, depending on the instrument and the experiment. And then in functional assays, excess dye can actually transfer to cells. So, for example, in the context of an EV uptake experiment, you may then get fluorescence coming from the cells that's not EV derived, EV specific. So after labelling, Vesi-SEC can be used as a fast cleanup step to remove that unbound dye and antibody before the downstream analysis. So I've got a picture of my face here because this is actually a testimonial that I wrote before I was at the company during my PhD a couple of years ago. And I was demonstrating the use of Vesi-SEC for efficient unbound dye removal. And here I'd labelled EVs with a Bodipi maleimide dye, and then measured on a nanoflow cytometer before and after cleanup with Vesi-SEC, and also Izon's QEV columns, which is what I was using at the time. And you can see essentially this fluorescence baseline is elevated when there's no cleanup step, and you can only detect 54% of the fluorescent population here. I appreciate it's quite small, but after cleanup, the baseline drops in both cases, and now we're detecting between 66 and 68%. So in terms of performance, these 2 examples here are equivalent, Vesi-SEC's equivalent to other gravity-fed SEC columns. But the real benefit is that it's non-dilutive. So whereas here you can see I loaded 100 microliters a sample and retrieved 100 microliters, with the QEV, we loaded 500 and retrieved 3,500 microliters, so a 7-fold dilution there. So overall, yeah, showing that the performance is equivalent to gravity-fed SEC, but the workflow is massively sped up, and it's non-dilutive, which can be a real benefit for downstream assays. Another similar piece of work was done here by Christian Prusa from Philipps University Marburg, this time with an anti-CD63 antibody and a lipophilic membrane dye. And you can see a similar trend here. Instead of looking at the fluorescence baseline, we're seeing actually an increase in the population that's detected post cleanup. And increasingly, we're seeing more published articles using Vesi-SEC for both EV isolation and also cleanup for downstream analysis. So Vesi-SEC is very much not limited to one sample type or one type of downstream assay. It can be used wherever removing small contaminants or free reagents or reducing background signal is important. Another key challenge is plasma-derived EV isolation. So plasma is obviously a very important sample type for EV research, especially for biomarker discovery and clinical studies, but it's also a very complex biofluid. It contains obviously a high abundance of soluble proteins, but also lipoproteins, which can overlap with EVs, both in terms of size and density. So that makes them quite challenging to separate using conventional methods. So introducing one of our newer products we launched a couple of months ago. Essentially, it's designed specifically for plasma-derived EV workflows. And the workflow starts by mixing the plasma with the Vesi-Omics reagent, incubating for 30 minutes, and this selectively precipitates out lipoprotein, specifically low-density lipoprotein, or LDL. You can then load the EV-containing supernatant onto, in this case, I'm showing Vesi-SEC. You can also load it onto your existing SEC workflows. And then after a 1-minute spin with the Vesi-SEC, you elute your purified plasma EVs. So this is combining selective lipoprotein depletion with SEC-based isolation. So overall aiming to improve the purity whilst keeping the workflow relatively simple and also non-dilutive. And the really key thing here as well is scalability. So Vesi-Omics gives you the opportunity to process multiple samples very quickly, simultaneously, and you're really only limited by the capacity of your centrifuge, essentially. So this massively speeds up the workflow and it gives you more time to focus on other aspects of the research project, whilst also being able to process clinical samples quickly while they're fresh and still in a nice condition. In terms of performance, Vesi-Omics is designed to achieve a high EV recovery whilst also obviously depleting lipoprotein contamination. So it achieves an over 80% yield in terms of particles and up to 98% of lipoprotein depletion, whilst being non-dilutive. And we've also analysed the Vesi-Omics isolated EVs by single molecule localization microscopy on the ONI system, which is a super res microscope. And this is showing essentially that the workflow is not simply depleting lipoprotein, but it's also preserving intact EVs for downstream analysis. So for plasma EV work, that balance is really important. We want to improve purity without sacrificing too much EV material. And moving on to the final part of this first section, which is EV stabilization and storage. Even after EVs have been isolated successfully, they can still be lost very easily, either due to handling or adhering to plastics, and they can degrade quite quickly in standard buffers like PBS. Their concentration and also functionality really depends on how they're stored and diluted, and this is a real issue because samples are quite often low concentration and or low volume. So even a small amount of adhesion to tubes or pipette tips can lead to quite a meaningful loss of EV material. So this is our Vesi-Safe preservation buffer that's designed to stabilize EVs both during cryostorage and also when working on the bench. So it reduces adhesion to plastic surfaces, and it's been demonstrated to work for over 5 years with EVs stored in the -80. And then we've got some data here just showing how big of a difference it makes when comparing diluting EVs. These are fluorescent EVs in PBS versus fluorescent EVs in Vesi-Safe. You can see that whole population there is lost after 24 hours. And this is available in a book article here. So I'm going to move on to now EV labeling, which is also really critical for many EV studies, especially when thinking about uptake assays or biodistribution studies, or various imaging and fluorescence-based quantification. But it's also one of the areas where artifacts can be easily introduced. So People often look towards lipophilic dyes when we're working with EVs because, of course, EVs are membrane-bound particles, and so therefore it's a useful way to distinguish them from non-membrane-bound particles. But many membrane lipophilic dyes work by associating with lipid membranes. And a key issue with that is quite often it's reversible. So conventional lipophilic dyes will reversibly bind to the EV membrane, which means that dye can transfer from EVs to cells, for example, or other membranes. And this, in an uptake experiment, could lead to false positive uptake signals, where a cell becomes fluorescent, but the fluorescence isn't necessarily EV specific. Many dyes also form micelles and or aggregates, which can be confused for EV particles depending on your setup. So really, we're aiming for a stable membrane label with slow loss of fluorescence and no transfer to cells. So the Vesi-Dye LMB range, these probes are designed to address this by intercalating across the lipid membrane bilayer. And this achieves two things. So any excess dye that's not intercalated isn't fluorescent. So when measuring EVs alone, the background noise will be nice and low. Also, this intercalation is irreversible, so it specifically labels the membrane bilayer and won't transfer to cells or tissues in a cell uptake or biodistribution study. So overall, the message is that stable, low background labeling is essential if we want to use fluorescence as a reliable readout for EVs and look at their interactions with, for example, cells. So this is just summarising the range of Vesi-Dye different probes that we have. So they all work the same, but they have different excitation and emission spectra depending on your experimental setup. So there's a lot of flexibility depending on your instrument and if you have other fluorophores as part of your setup. And if you need any assistance in choosing the Vesi-Dye that's most ideal for you, I'm very happy to help with that. But essentially, the dyes are water soluble, so this helps reduce micelle and aggregate formation, as the dyes can literally just be dissolved in water or PBS. And yeah, they intercalate into the lipid membrane, and they're also fluorogenic, as I already said. So this is a slide just showing the example of EVs, red blood cell derived EVs, labelled with the Vesi-Dye, one of the Vesi-Dyes, and labelled with DI8, which is another kind of lipophilic dye. And you can see, so it's very small, apologies for this, but essentially I've pulled out the percentages out of these plots here, the second one on the top row of each panel. And when comparing firstly DI8, when this is mixed with just buffer, so like PBS, for example, you can see that really we shouldn't be seeing any fluorescence here, but we've got 11% particles being detected fluorescent. So we're getting clear aggregation happening here, which could easily be confused with an EV sample that's been labelled. And when we look at the EV sample, so red blood cell EVs plus the DI8, we get about 30% of the population that's detectable. Whereas looking at the Vesi-Dye, we get virtually no fluorescence coming through, only 2% when it's just mixed with buffer, versus the labelled EVs, we're now detecting 53%. So it's a big difference in the detectability of these EVs and the background noise as well. And as with the Vesi-SEC, we're now seeing a few more publications coming out staining EVs with the Vesi-Dyes, so you can navigate to these as well if you're interested. So coming to the final section of the talk, so I'm going to talk about normalizing and standardizing EV/nanoparticle analysis. It's a challenging but increasingly important area because nanoparticle measurements, as many people will see, can really vary between instruments and also laboratories. So it's especially true for nanoparticles when we're trying to work and push towards the limit of detection of so many different instruments. And I think overall it's a relatively understudied area, but probably only because we're only recently starting to have access to appropriate reference materials. So I'll introduce some of these now. But first, what are reference materials? Why do we need them? They're essentially a control material. So it's to make sure we ensure that what we're measuring, what we think we're measuring, is what we're measuring. When we're measuring EVs and nanoparticles, due to their size, it's really important to ensure an accurate measurement, and reference materials allow you to control for this. So we need reference materials that mimic the behavior of the samples, but also have multi-parameter capability so that we can measure them across multiple different instruments in multiple different settings. And having appropriate controls in place is really important, obviously, for generating robust data and ultimately saving time, justifying to reviewers, why your setup is robust. But there's several challenges in the field to do with this. There's overall a lack of standardization, and as I mentioned, that might be because we've just not had access to appropriate reference materials. There's also a limited availability of biologically relevant reference materials, and conventional calibration particles that people often use, like silica and polystyrene, don't well represent EV properties. So for instance, the refractive index isn't equivalent. Refractive index is the way in which particles scatter light. So yeah, the best sort of most biologically relevant reference particle for an EV would be an EV itself. And so I'm going to introduce now Vesiculab's range of reference materials, each to suit different applications. So you'll notice that I've separated them into biological and synthetic reference materials, each serving slightly different purposes. We have extracellular reference materials on the left, which are derived from HEC293 cells. They're highly concentrated and they're stable because they're suspended in that Vesi-Safe preservation buffer that I mentioned earlier. And in the case of Vesi-Ref CD63, it's been fluorescently engineered so that it either has GFP or MNG, which is another type of fluorescent protein that's 3 times brighter than GFP, present in the lumen of the EVs. Vesi-Ref naives, basically the same, except that it's unlabeled, and so it can be useful as an EV positive control in various assays that you have already set up, for example, Western blot. And it can also be used to validate antibody staining strategies that you want to check and have control for. Vesi-bind is quite interesting because it has FC binding domains exposed at the surface. So that means that these EVs can be functionalized with an antibody of choice. And then we've also recently launched Vesi-Ref Rainbow, which is a polymer nanoparticle. It's a reference material that's fluorescent in multiple channels and very monodispersed. So it's useful for tuning fluorescence on instruments for nanoparticle detection. And then also we've got the Vesi-Ref LiPo, which are liposome standards that are fluorescently labelled, and there's a range of different excitation emission spectra available for those. So overall, these reference materials have a refractive index that's much closer to EVs. Obviously, the biological reference materials are EVs compared to other materials like polystyrene. And yeah, so this is essentially the range that we've got. And I'm going to talk a little bit now about these 3 in particular. I'm going to show some data for. So starting firstly with the Vesi-Ref CD63, these are engineered HEC293 derived EVs expressing CD63 tetraspanin fused to M neon green, which is the fluorescent protein. And they're suspended in Vesi-Safe buffer, so very stable in terms of both concentration and also fluorescence. And one key advantage is they can be detected in the FITC channel, so that makes them compatible with many different flow cytometers, imaging platforms, and whatever fluorescence-based system you're using. And because they're EVs rather than synthetic beads, they're also more biologically relevant in terms of their composition and, as I said, the refractive index. So this is particularly useful for nanoflow cytometry, where scatter-based detection is strongly affected by the refractive index. And we've done some general particle characterization of these Vesi-Ref reference materials. And you can see some fluorescence NTA here, so they're very bright. Individual EVs can be detected. Some transmission electron microscopy and video drop data. And in terms of the different applications that you can use these EVs for, so they're really useful for setting up instruments for EV detection and defining gating strategies if you're doing flow cytometry. They're also useful as a positive EV control, assessing concentration linearity, and also performing comparisons across different instruments or experiments across different labs. And I've actually just submitted with my colleague Josh Price from the University of Birmingham a paper that is comparing multiple flow cytometers using this reference material. So it's really useful for that kind of cross-platform comparison. And yeah, the overall aim with this is to give researchers a more relevant control material for EV analysis. So this is some work that was done before I joined the company with Unchained Labs, and essentially they've used their Leprechaun system to confirm the engineering has worked with these reference materials. So you can see that when probed with an anti-CD63 antibody and imaged on the system, the co-localization of CD63 antibody with the MNG fluorescent protein shows that we're getting approximately 75% of the EVs that are engineered. And it's at a really high concentration, so it's nice and easy to work with. You get a lot of material, 5e10 times particles per mil. It's quite concentrated for an EV material. So yeah, a similar bit of work was done here on the Cytoflex Nano instrument by Beckman Coulter. And this is actually part of an application note that we did with them, which is available on our website. And essentially it's showing a similar thing. When probing with a CD63 antibody, we're getting around 70% that are engineered successfully. And also just showing some data that was done on the CellStream imaging flow cytometry platform. And you can see here how, when comparing to the buffer control, you can really nicely set a gate for those fluorescent EVs ready to then run your own samples. You know exactly where to look, essentially. So yeah, that can be really useful for setting up an EV gate. So there's a couple of publications here as examples where the Vesi-Ref CD63 is being used, both as a control material and also to study biodistribution. So because the MNG is luminal, it's bright, and its fluorescence is maintained when the EV is intact. And then let's talk a little bit about Vesi-Bind. So Vesi-Bind, as I said, is quite interesting because it's got several Fc binding domains at the surface of the EVs, and this means that it can bind antibodies. It's also got intraluminal MNG, so it's fluorescent as well. And some of the applications, example applications of this is quantification of EV antibody interactions. So you can assess how antibodies bind and assess the epitope accessibility on individual EV populations. You can also do some EV surface functionalization. So if you're doing experiments like antibody guided targeting or biodistribution studies, and linked to that, you can also optimize the antibody loading of that. So this is comparing an IgG antibody, the binding of that antibody to Vesi-Bind versus Vesi-Ref. So we're sort of using Vesi-Ref CD63 as a negative control here to see that that IgG is only specifically binding to Vesi-Bind. And it's also showing at the same time that Vesi-Bind still has intact CD63 tetraspanin epitopes that are available to the antibody as well. And so one example publication where this material is being used was only published last month, and it's looking at the quantification of EV antibody interactions using feeder or flow-induced dispersion analysis. So you can check that article out as well if you're interested. And then now I'm just going to talk a little bit about one of the synthetic reference materials that we've just released, Vesi-Rainbow, which is a multi-channel fluorescent reference material designed to support calibration and validation of instruments used to detect nanoparticles such as EVs, but it could also be LMPs, liposomes, viruses. So it's a really useful it's got a wide excitation and emission spectra. So it's fluorescent across multiple channels, making it very versatile. It's also monodisperse, around 140 nanoparticles. And unlike EV reference materials, whilst they're more biologically relevant, this material is more useful for instrument tuning. So, for example, laser alignment, optimizing the fluorescence setup of an instrument like laser power and thresholding, so that you have an optimal nanoscale detection setup. And it can also be used for comparing data measurements across different instruments and platforms. And this is just some example NTA data, just showing the emission in different wavelengths. And I've gone through the applications already. Here's some example data as well that was given to us by Andre Gorgons from the Karolinska Institute. And essentially, this is a flow cytometry example trace of the beads done on the Beckman-Coulter Cytoflex Nano. You can see that the beads are easily distinguishable from the background noise, this population on the left being the noise and box being the gated beads. And essentially what he's done here is shown against every other fluorescence channel, the fluorescence of the beads. So we're spanning between 500 and almost 800 nanometer wavelength here with the excitation, with different emission with different excitation lasers used. So overall, the main message here is this can be a universal QC bead for multiple fluorescence instruments such as flow, NTA, etc. And it's very economical because you really don't need a lot. This was diluted to 1E7 particles per mil in PBS, so less than a microliter technically that you would need to do this. So hopefully I've given a good overview of all of the different reference materials that we offer, some of their different applications depending on your experimental setup. So to summarise, the main message from today is that EV workflows are only as robust as their individual steps. And here at Vesiculab, we're trying to provide end-to-end support across the entire workflow. So firstly, faster EV workflows increase efficiency and scale and reduce material loss. And secondly, stable EV labelling is essential for reliable analytics and functional assays such as cell uptake. And thirdly, EV reference materials are increasingly important for assay validation. They allow for more reproducible and interpretable EV measurements. And across all of these areas, the goal is the same. We want to help preserve sample quality and improve the confidence in the final data set. So yeah, that's kind of a whistle-stop tour of everything that we offer. And I guess now I'll welcome questions. So if you've got any questions, feel free to put into the Q&A chat and I'll check them now. If you think of any questions after the session, my email's here, like feel free to note it down and get in touch. Very happy to chat, whether that's kind of more specific questions related to your specific experiments or just general questions about how the products work or how to use them, then yeah, do get in touch. So I'll stop sharing my screen, and just as a reminder, the slides will be available after this. We're going to share the webinar link for everyone. So I probably whizzed through that quite quickly, so apologies if that was a